the Biomarker network. We had, I think, a really successful Monday session, and today we're going to be focusing on presentations looking at biomarkers in relation to different health outcomes. And as Eileen said, I think on Monday, this really represents, I think, and reflects really well the growth in this network over time, and in so doing reflects the growth in work looking at these biomarkers in relation to health outcomes. So it's pretty exciting to see not only the research growth, but just how many people are involved in this now. So we welcome all of you, and we're thrilled that this sort of virtual meeting seems to be working out as a forum, since it seems likely that we will be continuing to meet in this manner for at least a bit more time. We have 4 presentations today. We're scheduled to go 2 hours, and each presenter has been given 15 minutes for their presentation, and then we have 5 minutes after each presentation for people in the audience to ask questions or points of discussion. I think we've made everybody a panelist, which means you can hopefully raise your hand when we get to the point of entertaining questions. And then Eileen and I will try to keep tabs on who would like to speak and let you know that it's your turn to ask your question. So I think we'll go ahead and get started. Our first presentation will be by Cathal McCrory, and he and his colleagues are presenting on lifespan adversity and epigenetic age acceleration in the Irish Longitudinal Study of Aging, otherwise known as TILDA. And Cathal, the floor is yours.
Thank you, Trisha. So hopefully everyone's able to see my slides.
Yep.
It looks good.
It has, there you go.
Good.
So I guess I just want to begin by thanking the panel for their invitation to speak today. I guess some stage I'll actually get to meet you all in person when I get to one of these biomarker meetings, but it wasn't to be this year. So I also just want to acknowledge my co-authors in the paper here, who's Giovanni Fiorito, Paulo Venus, and Roseanne Kenny. So to begin, numerous studies suggest that the experience of trauma and adversity is associated with increased risk of chronic disease and psychopathology. And these adversities include different forms of abuse and neglect, family dysfunction, parental loss, separation and divorce, experience of war, and also socioeconomic adversity. And I guess this work is most closely identified with the research of Vincent Fleddy and his collaborators on the Adverse Childhood Experiences study in the US, but they're not the only ones who have an interest in this. So this paper here shows data using the TILDA study, which examines the relative risk of having a doctor-diagnosed disease in late adulthood by the experiences of different types of adversity during childhood. And you can see here that the experience of adversity during childhood, particularly physical abuse and childhood sexual abuse, is associated with increased risk of having these various disease types, net of a whole range of other confounding factors. And it's also associated for each of the adversity types with increased risk of comorbidity of different disease types. But I guess for the biomarker group, and more fundamentally, the question for us is, how do these pernicious social influences get transduced at a more fundamental biological or molecular level to hasten the aging process? And in recent years, the idea that epigenetic mechanisms may partly explain the persistent effects of adverse childhood experiences has been gaining some traction. However, a meta-analysis by Wolf et al. In 2018 suggested that the evidence to date is inconclusive. So some studies showed effects of adversity on epigenetic age acceleration, and others showed either null effects or some pointing in the opposite direction. And I just want to quickly digress into what epigenetics is. Now, I'm a psychologist rather than a biologist, so there's probably people more qualified to explain this than me. But in essence, epigenetics refers to chemical modifications to genes that change the way in which genes are expressed without changing the underlying structure of DNA. And you can think of this as a change in phenotype without a change in genotype. And the process most widely studied in the context of aging is DNA methylation. Now, levels of DNA methylation are closely related to how well DNA is transcribed into RNA. So as levels of methylation increase, you get less and less gene expression until the gene is silenced or repressed. And I guess this process is illustrated graphically here in this graphic, which I took from a paper in Child Development. So you can see here that these cytosine phosphate guanine sites are heavily concentrated in the promoter region of the gene. And here you can see the open circles indicate that the CPG site is unmethylated. That means that you get access to transcription factor binding sites, and therefore you can get gene expression. In the bottom panel, you can see that these CPG sites are methylated. They cannot access the transcription factor binding sites, and therefore you don't get gene expression. Now, a few years back, some investigators established that levels of DNA methylation at a small number of CPG sites across the human methylome were highly predictive of chronological age. And the regression of DNA methylation age here on the y-axis on chronological age is hypothesized to represent a measure of biological age acceleration. So a positive residual signifies someone aging faster than their chronological age, and a negative residual indicates someone who's aging more slowly than their chronological age. And the interesting thing about this too is that the epigenetic clock does not run at a constant rate. In fact, it's suggested that it runs fastest in early life. And this makes sense intuitively as well, because we know that development is nonlinear. So some people then again suggest that this might represent a mechanism through which these early childhood experiences have greater impacts than they might have at another stage in the life course. So Marini et al., a paper using the ALSPEC cohort, examined the relationship of 7 different types of ACEs measured between 0 and 7.5 years of age with epigenetic age acceleration in that cohort. And they found that financial hardship was the only adversity that was associated with age acceleration. And that's when they used an ever versus never dichotomy. However, when they looked at sensitive period effects, they found that physical abuse at 3.5 years of age, financial hardship at 7 years of age, and neighborhood disadvantage at 7 years of age was associated with about 1 month of epigenetic age acceleration. A separate study involving the same cohort looked at the 10 ACEs prospectively measured between 0 and 14 years of age. And this time they found that emotional and physical abuse was associated with Harvath age acceleration in girls, about 1.2 years of age acceleration, but not in boys. And again, they found evidence of a dose-response relationship between account of adversity and age acceleration in girls, but not boys. So just moving to the present study, what we want to do here is examine whether the experience of lifespan adversity is associated with epigenetic age acceleration in the TILDA cohort. And secondly, if it is, we want to try and understand the pathways through which adversity precipitates age acceleration. So TILDA is a member of the HRS family of studies, and it involves a sample of 8,500 community dwelling older adults aged 50 years and older at the time of recruitment. And just on the map here, this is the Republic of Ireland. It's missing its head there in Northern Ireland, but these are all the district electoral divisions from which we drew the sample. So you can see that we have a really representative spread across the country. And in fact, I think our sampling fraction is about 1 in 150 of all older people aged 50 years and older in Ireland. Our data collection is very similar to the HRS family of studies. So the baseline sweep of data collection occurred in 2009. Respondents completed a computer-assisted personal interview, a self-completion questionnaire. It was paper and pencil leave behind in the household, which contained more sensitive questions, which I'll discuss today. And then probably one of the unique features of TILDA is the fact that all participants are invited to attend a health assessment centre where they do a detailed, comprehensive clinical health assessment administered by trained nursing staff. So you can see we have a health assessment in waves 1, 3, and 6 of the survey. And, but we also go back to them every 2 years. And the data I'm going to show you today is from the baseline survey, which was carried out between 2009 and 2011. I'm going to be using only a subsample of the TILDA cohort for whom we've got epigenetic data. And the subsample was selected based on the cross-classification of the respondents' father's social class and their own contemporaneous social class. We selected 4 groups based on patterns of intergenerational social class mobility. So we have a group who had enjoyed high childhood socioeconomic position and high adulthood socioeconomic position. Another group who had low childhood socioeconomic position and low adulthood socioeconomic position. And then we also have these upwardly and downwardly mobile groups as well. For my dependent variable here, I kind of already explained how the epigenetic clocks work. But for this one, I'm going to use the GrimAge clock, which is Steve Horloff's recently developed 2nd generation clock. And it was calibrated by identifying DNA methylation-based surrogates of 12 plasma proteins and smoking pack years as well. And having done that, they then regressed time to death on those methylation patterns that they had, and they identified about 1,000 CPG sites that jointly identified mortality risk. And in some subsequent papers, one that I've been involved with myself, we find that this GrimAge epigenetic clock is a strong predictor of health span and lifespan. In fact, we found that a 1-year increase in GrimAge age acceleration increases the hazard of all-cause mortality by about 18% over a 10-year follow-up period in the TILDA cohort. We used a life events checklist to assess lifespan adversity. So we have 5 childhood adversity types that occurred before the age of 18 years. So we have death of a parent, repeating the school year, parental alcohol or drug abuse, whether they're physically abused or sexually abused during childhood. And then we also have a number of mostly adulthood adversity types here, experience of a natural disaster, whether your spouse partner is addicted to alcohol or drugs, and so on. And we also supplemented this by 2 additional questions, which asked about socioeconomic adversity in childhood, which was a question which asked them to rate the circumstances of their household growing up, very well off, average, or poor, and poor was described as poverty. And we also asked them to indicate whether they own their own home or not. And this is a very good indicator for us in TILDA, because about 80% of people in Ireland own their own home, and almost all of the older person's assets in Ireland is bound up with the house value, essentially. This shows here the percentage of the sample reporting each adversity type by sex. And you do see some differences here in reporting. So for example, you can see that men were more likely to repeat a school year, but women, by contrast, were more likely to experience childhood sexual abuse, about twice as likely. Men were more likely to report childhood poverty, but you can see here that women, for example, are more likely, or sorry, men are more likely to report serious illness, but women are more likely to report not owning their own home. Now this next slide shows the age and sex adjusted independent association of each of those adversity types with epigenetic age acceleration as indexed using the GrimAge clock. So on the y-axis here, we have the constellation of childhood adversities, and middle panel here, we have the adulthood adversities. And then at the bottom, what I did is I calculated the count of childhood adversities, a count of the adulthood adversities, and then we also have a count of the lifespan adversity, which is the sum of childhood and adulthood adversities. And then on the x-axis, we just have age acceleration in years. So if you lie to the right of the 0 axis, or the 0 point here, it indicates age acceleration. And if you lie to the left, it indicates age deceleration. So I'm just going to show you the results here for the childhood adversities adjusted for age and sex. So you can see that the death of a parent was associated with 1.3 years of age acceleration. Repeating the school year counterintuitively is associated with epigenetic age deceleration. And you can see big effects here as well for childhood poverty, so associated with 2.1 years of age acceleration. And we also see effects in adulthood as well. So experience of a natural disaster, having a spouse, partner, or child who was ever addicted to alcohol or drugs, and not owning your own home were all associated with significant epigenetic age acceleration. And finally here then you can see that the counts as well are all associated with significant epigenetic age acceleration, ranging from about 0.5 to 0.7 years per each adversity experienced. We also tested for effect modification by fitting separate sex by adversity interaction terms. And the way to interpret this chart is if essentially if there's no difference, as you see here, for repeating the school year, if it's small, that means that there's no difference between males and females. If it lies to the right, it indicates that the adversity is associated with greater age acceleration in men. And if it lies to the left, it tells you that it's associated with greater age acceleration in women. And what you can see from this is that none of the individual contrast was significant. However, we did note that the experience of childhood sexual abuse was associated with greater epigenetic age acceleration in men compared with women. So it was about 2.7 years more for experiencing childhood sexual abuse from men compared with women. And you see in the opposite direction here, the loss of a parent during childhood and childhood poverty seems to have bigger impacts on women compared with men. I said to you earlier, if we find that some of the associations were significant, we try to see what explains that away. This is by no means a complete list, but it's a list of potential mediating variables. And what I just want to show you here is the association of each of these putative mediating variables with grim age acceleration. So you can see that compared with the tertiary educated, the primary educated are about 2.6 years older epigenetically. You can see huge effects here for smoking. So compared with never having smoked, those who are current smoker and smoked for 30 years or more are about 8.5 years older epigenetically. You can see that physical activity is protective, that hazardous drinking is associated with age acceleration. But you also see something interesting here going on with the psychosocial variables. So control refers to the belief that you can exert influence over your social environment to bring about desired outcomes, is associated with a significant age deceleration. And again, having a larger social network for each additional person in your social network is associated about 1 year of epigenetic age deceleration. And you can see increases here for depression and for perceived stress as well. So I'm coming to my last set of slides. So what I want to do...
You're at 15 minutes, so you should wrap up.
Okay, I'll do really quickly. So
okay, thanks.
Okay, what I just want to show you here is, this is the slide that we showed earlier that showed the significant associations. And what I want to do here is just show you how much attenuation we get as we adjust in turn for education, then education plus the behavioral risk factors, then education, behavioral risk factors, and the psychosocial factors. Okay. And you can see there that when we adjust for education and essentially smoking, that you see a very large attenuation of the coefficients. And the only two that remain significant in the final models is experience of a natural disaster and experience of a life-threatening illness or accident. So to summarize, childhood adversity was not strongly related to epigenetic age acceleration, with the exception of childhood poverty. Adulthood adversity was more strongly related to epigenetic age acceleration, but that may be because the events are more proximal to when the assessment of the outcome occurred. Psychosocial processes may be implicated, but there's more work to be done here. And the message to take away this is that smoking is really, really bad for you. And because I'm under pressure to finish up, listen, I guess people during the discussion can tell me what they think the strengths and limitations of the analysis is here, but I'll just leave that screen to finish. Thank you.
Thank you very much. Really interesting. So the floor is open for questions. or you can put questions in the chat. Eileen and I will try to keep an eye on
put up
both.
your hands and
Yeah.
Can I ask a question? Because I cannot see how to raise the hand. I think we're still on.
You put up panelists, attendees at the participants, and then to the right, there's a thing that says more, a few dots, and there's a raise hand about halfway down. At least that's what it is on mine. I'm the host. So.
Oh, yes, I found it. Thank you. Can I ask a question? Is that okay? Hello, thank you very much for this very interesting presentation. Paola Zaninotto from UCL. I'm one of the managers of the ELSA study. I wanted to ask you a question about your model for the cumulative adversities.
Yep.
So you've shown that some of these were associated. If you can go back to I think it was the first slide where you got your chart.
This one?
Yes. So this one is adjusted for age and sex.
Yeah.
So I wanted to ask you two questions. First of all, each of these cumulative adversities, were they entered in separate models or they're mutually adjusted?
They're mutually adjusted here for age and sex. Oh, so do you mean the adversity types? Were they mutually adjusted to the adversity? No, but I can show you that because I was anticipating that question. So there it is.
Oh, so this is when you put them all together in the model.
So age and sex and simultaneous adjustment for the other adversities as well.
Okay, and the other question I had was about whether you've adjusted those for all the other variables that you've included in the other models and what happened with the attenuation there.
So yes, I have done that, and not a lot changes compared to the final set of models that I showed you. So essentially what happens is death of a parent is correlated with childhood poverty. So once you adjust for childhood poverty, death of a parent falls out.
Okay.
Yeah, but you still see experience in a natural disaster and what was the other one? A life-threatening illness or accident were still associated with age acceleration, even in the final set of models.
And when you adjust it for education, CASP-19 and all the variables, so do you see a reduction, or is it more or less the same?
No, so sorry, I had to speed through those models, I know. So, and let me just adjust my screen. So the reason why we adjusted for education in the first instance is because TILDA has a different confounding structure for some of the adversities compared to other data sets. So for example, individuals in TILDA who are more advantaged are more likely to report physical abuse and sexual abuse. So that's why we adjusted for education on its own there in the first instance to see if there's any suppression of the effect, but we don't see that. And you can see that the effect of childhood poverty is reduced by 27% when you adjust for education on its own. But then when you adjust for education and the behavioral risk factors, that association of childhood poverty with the age acceleration is reduced by 70%. And almost all of that happens. I've done a decomposition analysis, and it's all happening through smoking, essentially.
Okay. And that's the same for the cumulative adversities, is it?
Yeah, you see the same thing with the count. Yeah, the count of the cumulative adversities is the same thing. I left it out here because I didn't think I'd have time to get through them all, which I didn't. But yeah, so you see the same thing when you adjust for education and smoking on its own. You explain more than 50% for the count of childhood adversity, for the count of adulthood adversity, and the count of lifespan adversity as well.
Thank you.
I just want to make sure.
Do we have any other questions here?
Yeah.
Anybody else? I'd just like to ask, so Katal, we thought this was progress because when GrimAge first came out, it's so much more highly related to the dependent variables we're interested in. But now that we find out what that the way it's been developed based on smoking, and it's so controlled by smoking, I wonder if we're better off at uncovering underlying processes of aging versus the fact that you concluded very nicely smoking is bad for you, but we did kind of know that without doing methylation.
No, I agree. And in fact, if I can just go back to one of the screens, when I spoke to Roseanne Kenny about this, we were laughing. We were saying, it's the putative mediating variables. Like, I mean, this is nothing new.
Right.
You know,
Well, and we've, in HRS, we've also fit another new DNA age marker, and that one is really telling you obesity is bad for you. And I'm beginning to wonder if we're starting to go in circles instead of uncovering these basic processes of aging that we're interested in.
Yeah, it comes back to one of the points on the last slide when we talk about different levels of analysis here.
Yeah.
So for some people, they're content to say that, well, there's an association between childhood adversity, the experience of an event, and then this outcome here, be it,
Right.
you know, cancer or heart disease, whatever it happens to be. And then other people are looking at, well, what's the pathways through which that's explained?
Right.
Is it smoking? Is it obesity? And then there's other people are thinking about what are the more fundamental processes through which it happens. So we're looking at this through different levels of the same lens, I guess.
So I also,
Is this Jin Cook Lee has
Yeah.
her hand up?
Yeah.
I thought you were at NIH, Jin Cook. Okay.
Oh, sorry, this is Jorge. I have.
Oh, okay.
You have a mutated gene code. Sorry, I am from Italy. Sorry, hello.
Right.
Everybody.
Okay. You're. Okay.
I'm not an expert, but just by listening to this last discussion, I would say that the new side of this is the relative effect. So you could have said before that smoking was bad, but now you can say it is worse than these other effects. So I think, which you didn't know before.
Okay. Thank you.
Thank you.
Teresa, do you want to?
okay, I think if there aren't any other urgent questions, I think we'll move on to our second presenter. Giorgio Di Gesa will be presenting on associations between changes in vitamin D levels and health in later life using evidence from the English Longitudinal Study of Aging. And Giorgio, the floor is yours.
Hi, morning everyone. And hopefully you can see the screen. And you can
Yes, we can.
Brilliant.
There you go. Perfect.
Excellent. So, again, good morning or good afternoon, depending on where you are. And thanks for inviting me here. It's a shame we couldn't meet in person, but, fingers crossed next year. I am a fellow at UCL, and today's presentation is on the associations, and as you've mentioned, which we
Giorgio, you're going in and out.
Oh, okay. Hopefully, is it better now?
Yeah.
Okay.
Yes.
I'll try to stick to have the mic next to my mouth.
Yes. Thank you.
And this is work that together with colleagues at UCL. So standard outline of the presentation. So I'm going to give a little bit of a background and aim and objectives. I'll describe the data used for this study, and then briefly, some selected results and conclusion. So we mostly get vitamin D from sunlight exposure, and to a very smaller extent also from foods such as wild oily fish and egg yolks. However, vitamin D absorption declines with age, and recent studies suggest that about 40% of older people in Europe are vitamin D deficient. And this is surprisingly, at least to me, who am from Italy, mostly so in Southern European countries. Vitamin D is important for a number of health outcomes, and there is a large body of evidence suggesting that the deficiency in vitamin D is associated with both muscle and bone-related deleterious outcomes such as osteoporosis, poor balance, and muscle weaknesses, as well as with other disorders ranging from cancer to high blood pressure and depression. However, especially on the non-skeletal illnesses, the evidence at the moment is predominantly cross-sectional and hasn't really focused on changes in vitamin D levels and on older people. So in our study, we tried to examine and look at the relationship between vitamin D levels and health in later life, using longitudinal data, focusing on changes in vitamin D levels, and using a nationally representative study of older people. In particular, we used, as it was mentioned in the title, we used the English Longitudinal Study of Aging. That is an ongoing nationally representative survey of English respondents age 50 plus who reside, of respondents 50 plus who reside in England. And in this presentation, we use data from wave 6, 8, and 9. And our first wave was wave 6, because this is the first time that vitamin D levels were measured. And generally, up to wave 6, a nurse visit took place in the ELSA study every 2 waves or every 4 years. But from wave 8, and that was on the whole sample, but from wave 8 onwards, this actually was carried out every wave, so the protocol has changed a bit, but only on half of the sample at each wave. So to increase our analytical sample size, we decided to look at changes between 6 and 8 and 6 and 9. And in particular, we restricted our analytical sample to those respondents who had had a nurse visit at wave 6, which I refer to as our baseline measurement. And then at either wave 8 or wave 9, which would be like, our follow-up measurement. They also had to have their blood taken at both visits, and that's because some people might have not been eligible for their blood samples to be taken, or they might have refused to give written consent. And then, finally, they had to have a valid measurement of serum 25 hydroxyvitamin D concentration. That is, the best indicator of vitamin D nutritional status. So, with all these restrictions, we ended up having a sample of roughly 3,500 respondents. As for our main variable of interest, that is changes in vitamin D levels, according to most of the advisory boards, such as the Institute of Medicine, as well as the European Food Safety Authority, there is a general consensus that levels above 50 nanomole per liter of the serum 25 OHD concentration should be considered as sufficient levels of vitamin D. Anything below 50 is considered insufficient, and some bodies actually suggest that we should further distinguish between levels below 25 or 30 nanomoles per liter and consider these ones as severely deficient. But for this presentation, I am only going to be looking at the cutoff of 50. And therefore, given we've got 2 waves and a binary variable for vitamin D levels, that means we actually looked at 4 potential changes over time. That is, respondents could have a sufficient level of vitamin D at both waves, or an insufficient one at both waves, or they could actually swap and change categories. So, become at risk. So, having insufficient levels of vitamin D, or no longer having this insufficiency. And I'm not going to go into this, but I just also want to let you know that we try to do other checks and other measurements of changes in vitamin D levels, including the more subtle 3 category, so including the severely deficient ones, and looking at these 9 potential changes, including we also looked at times that people were at risk, so if at both way or at either waves. And then we also considered the 3 category baseline levels of deficiency or levels of vitamin D, and then we looked at also absolute changes over time. But for this presentation will just stick to the 4 category changes. For outcome variables, we've looked at different families of indicators of well-being, markers of inflammations, and muscle function. But again, for this presentation and given the amount of time that I was given, I will mostly just present data on depression, which was measured using the CESD depression scale, and we distinguish between those with 4 or more symptoms classified as depressed and those who had fewer symptoms. And then we looked at these 3 inflammation markers, and we distinguished between those who had abnormal levels of C-reactive protein, fibrinogen, and white blood cells, and those who had levels below well-known thresholds. As covariates, we included the usual suspects, so some socioeconomic and demographic characteristics such as age and wealth and marital status, as well as a number of health behaviors and health variables, including waist circumference and chronic conditions. And of course, given the importance of sunlight exposure for vitamin D levels, we also included variables accounting for the season when measurements were taken, and also an indicator that would suggest if people were interviewed roughly in the same seasons across waves. The analyses were done with a series of logistic regression that would subsequently adjust for the covariates that I've shown you. And we also ran some robustness checks, either focusing on those people who were interviewed in the same seasons across the waves, and also adjusting, instead of just for the baseline characteristics and covariates, also for changes in covariates over time. So some descriptives. This little table graph on the top of the slide shows the changes that occurred among these ELSA respondents. And as you can see, that about 37% of respondents actually had insufficient levels of vitamin D at both waves, with just below a third of respondents actually having sufficient levels at both nurse visits and times of interview. And pretty much the same proportion, that is 16–17% of people who actually swapped categories over time. And if we just look at the unadjusted associations of these changes with the measurements, with the outcome measurements, that we're focusing on, generally, those who actually had low levels of vitamin D across waves were much more likely to be depressed and to have abnormal levels of the inflammation markers considered. When we look at the adjusted models, the situation doesn't change much for some outcomes, but for others it becomes a bit more messier, let's say. So this table actually shows the results for 3 selected outcomes, that is depression, abnormal CRP levels, and abnormal fibrinogen levels as well. And here I'm just showing 3 models, where model 1 controls for baseline characteristics of all the covariates that I presented earlier. Model 2 further adjusts for both baselines, so baseline depression, baseline CRP levels, and baseline fibrinogen levels, as well as for seasonality. And then finally, the third model repeats pretty much what we did in model 2, but it restricts the analysis to only those who were interviewed in roughly the same seasons. That is, winter and spring, or summer and autumn. And whereas, for depression and CRP, it looks like that those who had low levels of vitamin D at both waves were more likely to be depressed and to have the, abnormal CRP levels. For fibrinogen, the situation is a bit less clear, so maybe some ideas of what's going on could be helpful. Because it looks like, that those who experience one vitamin D deficiency, either at follow-up or at baseline, were more likely to have abnormal level of fibrinogen compared to those who had never experienced this across waves. So in conclusion, I think we can say that, that older adults with insufficient levels of vitamin D across waves and across the two waves considered were more likely to be depressed, and also, more likely to report some levels of the inflammation markers considered. Although, with fibrinogen, we're not sure what might be going on. Whereas, we didn't show you any results for muscle function, because in this case, we actually didn't find any associations with the two variables that we consider, that is walking speed and grip strength. And finally, this was a short presentation, and I probably also rushed it through a bit. But in general, for some of these associations, it looks like that once you account for seasonality, the associations become less strong, or the direction is there, but it's no longer significant. So that, it really suggests that seasonality plays a very important role in these changes of vitamin D levels across waves. And so that, perhaps having supplements could be a good way to ensure good levels of vitamin D, both throughout the year and throughout the life as people age. So I think that's it. So, thanks for your attention. And if you have any questions and comments and feedback, please, I'm all ears.
So the floor is open for questions.
I don't see any. Okay, Luisa?
Oh, yeah.
Yeah, hi, thank you for the presentation. That was really interesting. I have a question. So I start my video so you can see me. I was wondering, maybe I missed it. Did you check whether the depression in those people with high risk of depression occurred in both waves or only in the second wave? Because the background of the question is that I was thinking about the causality of the thing. So maybe people with depression behave differently, and they do not go out, so they do not synthesize vitamin D or things like that.
So I mean, that's a great question. Thanks. So if you just look cross-sectionally, again, it looks like, and there's been studies already published on this, that those who have low level of vitamin D are more likely to be depressed. But in our analysis, we both look at just at depression at wave 2, so at follow-up. Yeah. But then we also control for baseline depression. So we're looking at both just depression and in a way at changes in depression status. And so that, even if you control for depression at baseline, it still looks at those who had no vitamin D or insufficient vitamin D levels would be more likely to. But then, of course, it's a question as to whether, those who are depressed are less likely to also synthesize vitamin D. I don't know. It could be actually. I'm not suggesting any causality here. I'm very cautious about it. If I've mentioned causality, I apologize and take it back.
No, you didn't. You didn't actually. It was just my curiosity, because you have the longitudinal dimensions, so you have the possibility to at least see it a little bit. But that was my question. Yeah, you answered it. Thank you.
I think what we could further do perhaps is to include, to to maybe also control for depression the wave before the first measurement of vitamin D. You know, and actually that's something that we can do. So we'll do that.
Yeah, so maybe you can look at trajectories of depression and then.
Yeah.
Yeah. Thank you.
Thanks.
More questions
So there's a comment from Roseanne.
in the chat?
Yeah, in the chat, there's strong circumstantial evidence for an association between vitamin D deficiency and severity of response to COVID.
Oh, yeah.
Wondering if you might have any thoughts about that.
Well, you know, thoughts. I think it looks like that increasingly, indeed, there seems to be a list, you know, an association between vitamin D deficiency and COVID-19. I think ELSA is collecting data probably, you know, as of next week. They started, you know, collecting data and also symptoms among these respondents. So, you know, we might actually do some analysis as soon as this data is available to see if and whether anything comes up when the new data and new waves are available.
Yeah, I attempt a question.
Go ahead.
So, Giorgio, do you think that based on your data, it would be worthwhile to look at vitamin D as an adjunct treatment in trials treating depression?
I mean, that's a tricky question, you know, because again, I think a randomized controlled trial, you know, would be the best way to actually see if indeed there is a causal relationship between the two things. But I think, you know, in general, there are some studies suggesting that if you have too high synthesized levels of vitamin D, like, you know, through supplements, that could be deleterious for health. But I think it probably wouldn't harm, given that vitamin D is associated with so many good health outcomes. You know, it wouldn't harm to be more conscious and to perhaps supplement vitamin D, at least in those months when the exposure to sun is very limited, and especially, you know, given also, I think, you know, especially in Southern European countries where you would expect that vitamin D level wouldn't be so low, but, you know, with all the sunscreen, sun protections, you know, and the covering up, actually, even in the summer times, you know, people are not exposed as much as they should, or the recommendation looks like. And, you know, these are also countries where depression rates are higher than, you know, if you think of Greece and Italy and the shared data sets, you know, depression is higher here than other countries. So there might be an association. I don't think it would harm to have supplements of vitamin D. Whether that's going to cure depression, it's a different question. So I don't think we've got the answer for that.
I don't see any more questions, Tressa.
Okay. Well, then we will move on to our third presentation, which will be by Brenton Keller, and he will be presenting on the interaction of age and mild cognitive impairment on brain morphometry, neuroimaging analysis from the Longitudinal Aging Study in India, the LASI-DAD, Diagnostic Assessment of Dementia.
Excellent. Can you guys see my screen?
Yes.
Perfect, perfect. I'll get started now. Hi, I'm Brenton Keller, and as it was just said, my presentation will be on the interaction of age and mild cognitive impairment on brain structural morphometry, and these are neuroimaging analysis from the Longitudinal Aging Study in India, Diagnostic Assessment of Dementia, or LASI-DAD study. Specifically, let me move this out of the way. I have the little thing right here. The LASI-DAD study is a subsample of the LASI study, and thus far the LASI-DAD study has had 3 data phase collections with a total collection of 4,096 participants aging from 60 years and older. And the goals of this study were to develop a protocol to assess dementia and mild cognitive impairment, the prevalence of dementia and mild cognitive impairment in India, modifiable risk factors, and also the impact of dementia and MCI on the families and society. And for these 4,096 participants, the LASI-DAD testing includes a variety of cognitive tests, which go over domains such as executive functioning, orientation, language, memory. We have informant reports, including the JORM IQ, BLEST. We have physical measures such as blood pressure, and also self-report index such as activities of daily living, instrumental activities of daily living, nutrition, Beck's anxiety. Now, the MRI cohort was a subsample of the LASI-DAD study, and goals were to verify structural, such as morphometry and microstructure, and functional changes associated with dementia in the Indian population, with a comparison to Western population findings, along with also looking at novel variables, because we have a rather extensive bank of factors that we tested on all these individuals, looking at novel variables that might contribute to MCI through brain structure morphometry changes. Our testing was done at 3 facilities: NM Medical Mumbai, Institute of Neurosciences Calcutta, and NIMHANS Bangalore. We've also added Delhi, but that will not be included in these results presented today. And one thing worth noting is that compared to the overall LASI-DAD study, in the LASI-DAD MRI cohort, we undersampled those with more pronounced memory loss. And the reason why we undersampled those individuals is we wanted to longitudinally track how memory loss changes over time and how this might be related to structural or functional changes in the brain. So when we look at the sample differences between the LASI-DAD study and the LASI MRI study, we see that there isn't a significant difference. Can you guys see my mouse cursor when I move it like this? Yes. There is not a significant difference in age between the LASI-DAD overall cohort and the MRI participants. But when we look at factors such as education or cognition or HMSE, which is the Hindi mental state exam, which is a little bit different from the MMSE, but same type of concept. And also risk of dementia, what we see is that in our MRI cohort in general, we have significantly reduced chances of significantly reduced dementia and significantly increased cognition. But when we look at our 3 different sites, Calcutta, Mumbai, and Bangalore, we do not see significant differences in these cognitive and dementia risk factors. However, I do touch on later, there is a significant difference in rural living between our 3 sites, with Bangalore having around 56%, which is more representative to the entire LASI-DAD study. Also worth mentioning is how we defined MCI. We had a consensus between psychiatrists who looked at the different cognitive and informant reports and biological factors for each participant, and they had to reach a consensus as to whether or not the participant had a CDR rating of 0, 0.5, or 1, designating MCI or dementia status. And because we undersampled those with more intensive memory loss, we see, at least in our cohort, around 50-50 split of those that are CDR of 0 and those that are CDR of 0.5. So now to get into the actual brain morphometry, what we have here is on the right-hand side are all the brain areas, the cortical, subcortical, and high-resolution hippocampal areas that are included in our analysis. And for each of these areas, we have measurements such as volume, thickness, surface area, curvature details, which were produced by the software package FreeSurfer. What we're looking at in all these different cortical areas are changes associated with age, MCI, and the interaction between age and MCI while controlling for factors such as gender and intracranial volume. There's a lot of different things that we could have done with these models. For example, we could have done a machine learning type of algorithm to classify between the two, but given the aims of our study, we wanted to first focus on which brain areas show the significant differences between groups. So here, for example, on this slide, we're kind of showing the different cortical areas. And in terms of dementia risk, when we look at cortical areas, we're mostly going to be focusing on the temporal areas, and then subcortically, which is not shown in this slide, the hippocampal and ventricular areas of the brain. So there are some differences between our 3 MRI sites, and this is a section that is very much worth noting. At NM Medical Mumbai and the Institute of Neurosciences Calcutta, we use a 3 Tesla Siemens Skyra system, whereas at NIMHANS Bangalore, we use a 3 Tesla Philips Engineer system. All 3 systems use a 32-channel head coil, but because of the inherent differences in just how the protocols are entered into these different scanner manufacturer brands, differences exist in our data acquisition. At the bottom here, we have some representations of a T1 scan from each of the 3 sites, and what we can kind of see right off the bat is there is a little bit more whitening of an effect at the NIMHANS Bangalore location. So to harmonize between our different sites, we used a tool called Combat, or neurocombat harmonization, which is a commonly used tool in genomics. It removes intersite technical variability while preserving intersite biological variability by using empirical Bayes to improve estimation of the different features of the brain. So specifically, we use a data matrix that consists of all the volumes and thicknesses and surface areas for all of our structural MRIs. We input also where each subject is from, Bangalore, Mumbai, or Calcutta, and we also include a matrix specifying biological covariates of interest, in our case, age, gender, and disease status. And to see what this actually looks like before and after harmonization, in this graph, what we have here is each of these bars represents one subject. In the red, we have our subjects from Calcutta. In the green, we have our subjects from Mumbai. And on the right-hand side, we have our subjects from NIMHANS Bangalore. What's kind of noticeable from this graph, you'll see it more in the next slide, is that on the right-hand side, the individuals from NIMHANS Bangalore have increased thickness. So again, each of these bars represents one subject, and of these bars, it represents the 60, 70 plus cortical areas of thickness. So for example, on the far left-hand side, we have an individual who has brain thickness going from 2.2 to around 2.5. And what we kind of see here is there is a site effect for NIMHANS Bangalore on the right-hand side. This is pre-harmonization. After using the Combat neuroharmonization, we see a leveling out. It removes the intersite variability while still preserving some of the biological covariates of interest, which I'll talk about in the next slide. So just to go back and forth between the two, you can see how one of the ways in which we're focusing on making sure that our data between our 3 different sites are still comparable. So now when we actually look at brain morphometry side effects and differences between cognitively normal and mild cognitive impairment individuals, when we look at gross anatomical areas such as total gray volume or cerebral matter or total cerebral spinal fluid, we see that we do not have significant differences between our 3 sites. But when we look at factors such as the cognitively normal versus MCI individuals, we do notice a significant difference in both total gray matter and cerebral white matter. Now, when we break this down, one of the areas we first focus on is the ventricle volume or lateral ventricles. Here on the right-hand side, we have highlighted all the subcortical areas in the brain in light blue, and then the actual lateral ventricles are these dark blue areas right here. So the light blue areas represent all the subcortical areas, whereas the light blue represents the lateral ventricles. It's been shown in numerous studies that MCI and Alzheimer's disease, those individuals have increased lateral ventricle volume. And in our study, we show a similar type of effect with a almost doubling of size of lateral ventricle volume in 15 years for those with MCI. And we see a similar type of association or similar type of finding for hippocampal volume, specifically that there's around a 5 to 10% decrease in hippocampal volume over 15 years for those with MCI. Again, our results are only from a single session for each of these subjects, so these have not been tracked longitudinally, so there's limitations as to what we can actually say in terms of the degeneration of the hippocampus or the increase of ventricular volume. But ultimately what we're seeing is, as I just said, for like the hippocampal volume, we're seeing around a 5 to 10% decrease, or a 10 to 15% decrease over 15 years. Now we also have high resolution hippocampal scans. So what we can do is, instead of just looking at the hippocampus as a whole, we can look at the different subfields of the hippocampus and track which areas are really showing this degeneration profile. And what I'm showing in this graph is all the ones with a red border are significant areas showing significant change. So specifically, we had a simple linear model for each of these 12 different brain area, 12 different hippocampal brain area subfields. And we looked at the effect of age plus MCI status and the interaction between the two. Ultimately, what we're seeing is that the areas that are significant have been shown to have a negative beta. And these areas more specifically are the CA1 subfield and CA3 subfield, which has been pretty well documented in studies that have examined high resolution hippocampal changes with MCI status. But we also identified areas such as the presubculum and the subculum. One thing of note though is the extreme variability that we see in these MCI subjects, such as with the healthy control subject, we see a nice type of standard deviation around, you know, the different means for the different different hippocampal subfields. But we see not only a decrease in volume of these subfields for those with MCI, but we see an increase in variability. And what I think that might be due to is differences in how these brain areas are categorized, because there's starting to be this slight degeneration. So it could just be that some of these areas are, instead of it being classified as the presubculum, it's being classified as the subculum with degeneration. But overall, it's showing that there are significantly reduced changes of these subfields in MCI. And in this study, we're looking, rather than ventricles and subcortical areas, we're looking at cortical areas. And what we found is that there is a significant change in both the superior, the left and right superior temporal sulcus, and also the right middle temporal cortex. So we included the MRI coordinates, and these seem to be pretty consistent with what has been found in literature. However, unique to our study is how we did the age and cognitive status interaction. Lastly, I just wanted to touch upon the effects of literacy and what can be done with our study. So unique to our study is that around 57% of the LASI-DAD sample is illiterate, and factors such as male gender and younger age were found to have an increased proportion of being literate. But when we look at CDR ratings, we see that literacy, either directly or indirectly via an increase in general cognition, because those that were literate typically had higher general cognition, essentially literacy correlated with decreased CDR rating. So we see that literacy plays a part with, you know, the probability of having dementia. And previous studies have shown that the posterior corpus callosum plays a role in literacy in the brain. And on the box and whisker plot at the bottom right, it shows that when we only look at the CDR equaling 0 individuals, so this graph only shows those that are MRI with a CDR equaling 0, we see that those that are literate have increased corpus callosum posterior volume. However, when we bring in the effect of CDR equals 0.5, this significant difference, which has also been shown in previous literature, seems to disappear, such that when we look at CDR equals 0, we see a significant difference, an increase in corpus callosum volume. But when we look at those that have a CDR equaling 0.5, we do not see we see a significant difference in the difference between these two groups, suggesting that there might be some type of interaction going on with literacy or being illiterate and dementia prevalence. It's something that we're currently looking at, and I mean, we're still trying to decipher these effects.
Brenton,
yes.
You're at 15 minutes, so you need to wrap up.
Perfect. Perfect. So limitations in this study, we did have, compared to most other studies, a high prevalence of MCI. And as I touched on before, we do have a high rural sample at NIMHANS Bangalore, which is not measured or mirrored at Mumbai and Calcutta. And if you can imagine ruralness in India as being a very unique type of important thing for brain development, it's something that we have noted and are currently examining. But here's the reference works, and thank you.
So the floor is open for questions.
Celine, do you want to ask your question in person? She asked, were separate regressions run for each region of interest? Is it the case for the hippocampal subfields too?
Yeah, so for the hippocampal subfields, we controlled using family-wise error. So it was different regressions for each one.
Celine, do you want to talk? You have more.
Looks like it says she doesn't have a microphone.
Oh, okay. If yes, a linear mixed effect approach, where ROIs that are taken as repeated measures may be more appropriate to address multiple comparisons. I see. Sorry, I don't have a microphone. So what was the approach taken?
So we calculate, we did it in two different ways. One, we looked at what it would be if we did Bonferroni method. And the only one that passed Bonferroni's method was the CA1 and CA3. But when we looked at the family-wise error, then we also had the two other areas. So we included that, at least in this presentation, on the slide with 4.
Okay. Are there any other questions? So Brenton, can I ask, that first harmonization that you did, is there any effect of the fact that the sample from Bangalore, I think it is, was such a rural sample, the right-hand sample, the blue sample? And
yeah, so
initial measurement have just been different because they were so rural?
So I actually did this harmonization quite a few different times, and I tried to figure out exactly what it was doing to our data set. And in one of the ones that I'm talking about, I included the rural as being a biological covariate of interest, and another one as being as not including it whatsoever. And either way, one, it didn't seem like ruralness really affected the thickness differences, but two, it did not change these charts here. So it didn't change, for example, the interaction of age and cognitive status. It didn't change hippocampal areas or these conclusions that we made for the hippocampal areas. But one thing, but it was very interesting to see how it did affect, because I also looked at, just looking at like NIMHANS, for example, what the effect of ruralness did to the brain. There was a slight change in how that was all calculated, but it didn't carry over into the other analyses.
Thank you. I think this is so interesting because it's one of the first population studies with a relatively large number of MRIs. I appreciate your showing it to us. Any other questions?
Okay, so we will move on to our fourth presentation, which will be by Martha McClintock. And Martha's going to be presenting on biomeasures in the time of COVID-19, remote self-administration collection. And I think Sharon is going to run the slides and Martha will let us know when to advance.
Yeah, that sounds
Sure.
good. So first slide, please. Hope it works. Wonderful. So I called this Biomeasures in the time of COVID, a riff on love in the time of cholera, because we started this project not in context of COVID at all. Our funding was cut and we wanted to retain sample size. And so we decided to see if we could figure out a way to remotely collect biomeasures from our respondents, to have them do it themselves in their homes. And originally, it was going to be a smaller sub-study of our NCHAP project, National Social Life, Health and Aging. But now with COVID, we were planning to go into the field now, you know, or February. And what older person is going to say, sure, come into my house and get within 6 feet of me without a mask. So we're now redoing this and thinking maybe what I'm presenting here now as the results of pre-testing and piloting might in fact be the main way we collect our data, biomeasure data, as we go forward. So first, acknowledgements in the next slide. I wanted to acknowledge that this, back up one, please. I want to acknowledge that today is Juneteenth and we're working. I think it's really important for all of us to take some time to figure out a way that the institutions that we're part of has given us enormous privilege and how it's restricting access to everyone having access. So that's just a plug for the day. And given that we study health disparities, I think it's particularly important. So in the next slide is the traditional acknowledgements to make sure that we get that clear in case I run out of time. So next slide is the 5 domains of the biomeasures that we are collecting. Cognition, and given the previous talk, we are going from high-tech fMRI, MRI, structural MRI, to paper and pencil. We're measuring, it'll be the first study of all 5 classic senses. Body measurement to look at obesity, activity measurements, and then collecting biosamples and getting them out of the home into the lab. Next slide. So here's what we call our bio box. I kept trying to figure out how to do an unpackaging video for you guys and couldn't couldn't do it. I was going to bring this to the meeting and pass it around, but it's a it's a big box, but it's easy to pick up. You could see there's a handle in the lower part there. And in the box is a styrofoam for the biosamples. There's the bio booklet, which is key. That's the booklet that's standing up. And then the supplies for various parts of the test. And the idea was to make this attractive and to make it easy to do and to appeal to the volunteer who would like to do 7th grade science fair measures on themselves, to have an element of fun. So in the next slide is the cover of the bio booklet that has all of the instructions. In the next slide is again the list of the domains. And what we did in order to create the bio book and the bio box is we pilot tested and modified the protocols and did tests by the people doing the measures themselves and then repeating them, looking at the concordance, getting community-based participatory research, getting their involvement in what was easy, what was hard to understand. So this was a long process. Then in the next slide, this is a close-up of what the bio booklet looked like. And this is another thing that was really important to us, that it be attractive. It's beautiful, heavy stock with those tabs for the different domains running down the right. Spiral so that it lays out flat so it's easy to fill in. Next slide. We then in the lab studied 69 people recruited from the community around the University of Chicago. 46 of those, when they did this themselves, someone was there in the room and could give them guidance. But it wasn't like they were administering the test. They were just there observing, seeing what was hard, what mistakes were made. And then there were 23 where they were totally independent and did the whole BioBox test. Based on that, the whole test is about 53 minutes long. Okay, and next slide. Oh, and there was 100% scorable data. That, after all, that was what all that previous piloting was to get to. So then NORC, who's the National Opinion Research Council, did a field test and did a 50-minute phone interview survey. And then in the end of that, asked participants if they would be willing to receive a BioBox. They did consent over the phone. We FedExed the box to them, and 100% of them went out and 100% came back. So we were really excited with that success rate, thanks to NORC. In the next slide is part of the success was from having 3 phone calls. So after it was mailed out, we talked to them and confirmed that it arrived. A week later, call up and say, any questions, you're having any trouble, and then a prompt to return the BioBox if it hadn't come back. After that phone interview, they didn't want to talk to us. It was really interesting. That option was there, but yep, fine, got it, doing it, leave me alone. Okay, next slide. So this is the cognitive measure. We gave the MoCA SA over the phone, but there were 3 that had to be done with paper and pencil. So you can see here, we called them puzzles. We didn't call them cognition. Most people don't know what cognition is. It doesn't mean anything. So there was a lot of editing and layout and decisions to try to make it at a newspaper, 5th, 6th grade level of reading and comprehension. So in the next slide, this is a close-up of how we really pared away the words for the instructions and tried to be reassuring, saying, one sentence about what we're testing. Then it may be easy for some, hard for others, and then other directions. In the next slide is an example of the TRAILS test that was in the bio booklet. And you may notice a salmon color running down the right-hand side. The puzzle section had a tab that was this color, and everything within the domain was color-coded. So we tried to make sure the information was there 3 different ways: color, graphics, and words. Okay, next slide. 100% of those out of the 49 returned scorable data. The next domain is senses. And we measured, as I said, the 5 classic senses. These are the materials that were in the bio box. On the left to right, the smell pens, I'll tell you about that. Then that little spatula thing on number 1 is a taste strip. Touch wands, this required that they find a helper and have them touch sensitivity on their fingers. We printed them out a big vision chart that they could keep as a take home with a wall safe tape, and then a high-tech red string and washers that they used to drop under the vision chart and then stretch out to exactly 10 feet to standardize how far away they were. The next slide shows you smell. So you can't see the print, it's too small. But again, this is an example of an instruction page. Those are the 5 pens. They're like magic markers. And we ask them to uncap it and just hold it under their nose and wave it back and forth briefly. Another thing you'll notice at this, down at the bottom, see that logo of a old-fashioned movie camera? That's a link to a video where they can go online if they want and see a video of how to do it. So in the next slide is then the page they would fill out. And so for the first pen, there were 4 choices of words and pictures, like rose was the correct example here. So they just check the box and flip the page. The next section, this is the results. In terms of returning scorable data, we got very good results. We did have an optional hearing test where they would call up, we gave them a code and take the 15-minute test. And 50% of the people chose that option. But we also have self-rated hearing, and we'll have interviewer-rated hearing, which in another study we've standardized against what's called the shoebox method, and then the hearing test, which is sound versus noise in the background. So in the next slide are measurements of the body, weight and waist. That's pretty straightforward. Again, good compliance. Next slide. Now we get to the bio samples. Here is how to collect, tell them how to collect saliva. You can see in the lower left there, a woman demonstrating. So I'm pointing at my screen, but using the C there, the over C on the right as the guide for saliva, and it goes into the vial. And after they do that, they put it in a styrofoam box in their refrigerator to keep it cool until they ship it back to us. In the next slide is collecting vaginal swabs. or vaginal cytology. And this time we're using this device called the Evelyn brush. It looks a little scary. It did to me at first, like, what, with all this stuff? But in fact, the part that's actually inserted is the lower right. It's quite small with just teeny little filaments that sweep up the cytology. And there's instructions on how to do it. It's very much like using a tampon with an applicator or vaginal medication. In the next slide, this is how we had them store it and pack it up. So we had this styrofoam box here and put both the saliva and the Evelyn brush in it. And we had a freezer pack that they kept frozen. In the next slide, we have the instructions. Again, forget the words, you can't see it. But this is the page that tells them how to pack up your biosample box. So you need your frozen ice pack, your refrigerated styrofoam box, packing tape, and a large Ziploc bag. That's because of condensation, spillage, etc. And packing tape. So they put all that. There's a little picture of the packing tape there that we send them. It goes in the styrofoam box. They seal it up and send it back along with the rest of the BioBox. So the next one tells you that with 92% provided a saliva sample that came back, and of those, 96% are assayable. So in terms of saliva, we're getting these 5 analytes listed on the left. The vaginal sample is a lower rate. We're going to be getting 5 measures out of that sample. But interestingly, when we also did in parallel an in-person interview with an interviewer who showed them how the the Evelyn brush worked and they could hold it and all that kind of thing. It was a 73% return rate. So this is right in line with just who's willing to get this kind of measure. And the next slide is the last domain, and then is fitness. And we had this wonderful gene active watch that we called it a fitness tracker for the purpose of explaining what they're doing. They have to activate it, and then it records continuously until it gets back to us. So it has the capacity to store information for up to 8 days of activity and sleep, but also, how do you get someone to do chair stands? How do you get someone to do frailty measures? Chair stands, 6-minute walk, a 10-foot walk timed. And our actigraphy group came up with the idea on the next slide of using this while they do these measures of frailty. So there is the watch. We send them veterinary stretchy tape. It sticks to itself, very easy. They strap it on first on their thigh, and then they are good to go with the measures. In the next slide is an example, hot off the press, I got this yesterday, from what the data look like from that tracker. And the different colors are the X movement in the X, Y, and Z coordinates. So you get very characteristic signatures of different kind of activities. And so this person said they did 5 chair stands, and lo and behold, we can tell that they did indeed do them. Not only that, we can tell how long it took, and you can see how regular that pattern is, indicating that they weren't having problems with stability or balance. The next slide shows the 6-minute walk. Now, the way we had that is they transferred the fitness tracker to their wrist, wore it for 8 days, and at any time during that time, we had them do a 6-minute walk test. And the question was, how can you tell out of the week where that signal is? So they were wearing it on their wrist, and we had them wave their arm up and down 3 times. And you can see here in the bird's eye view of the entire walk, that signal, and then on the bottom, blown up the signal, you can see the start signal and when they started, and then you can see their pace as they're walking along, swinging their arm in different X, Y, and Z coordinates. So from that, next slide.
Martha, I should also let you know you're at 15 minutes. So.
Yeah, I'm just finishing up, thanks. So 87% returned the fitness tracker with data, and therefore we have scoreable data for all of these 4 measures, 100% of them, if the device worked and it was activated, 100% are scoreable. So in the next slide is the summary. We have developed a way of collecting 22 different biomeasures remotely with people self-administering them in their home. On average, there's a just over 90% return rate. And if we get it back, almost 100% is scoreable. So that's it. Low tech.
Thank you. That's so creative, Martha. I mean, you guys, that's just really wonderful.
Thank you.
Yeah.
So
I did take a course at MIT in neuroscience, and it was taught by Letvin, and we had to do neuroscience with a C battery, paper clips, and duct tape in case we didn't get funding.
There you go.
Wow.
So the floor is open
Questions?
if there are questions.
People are floored. Okay.
Louisa?
Louisa.
You might be muted. We can't hear
I
you.
see.
Yeah,
I see.
okay.
Sorry. So here I am. Thank you, Martha. That was really impressive. I'm quite impressed. I have a question. Maybe I misunderstood. Did you say they were the gene active on the wrist for like a whole day?
For 8 days.
For 8 days, yeah.
8 days.
So how do you know when they did the chair stand, not the chair stand, the 6-minute walk? Did they write down on which day? So how can you find data?
We did ask them to write it down, but sometimes they forgot. But that's why we had them wave their arms. And
Yeah.
That's
I understood that, but I know from my own experience that this is a lot of data, and you do not want to go through the 8 days to find a signal. So
this is done by hand to get this to show you today. This is but we're thrilled to know what the signal looks like, and so we can program searching for that.
Okay.
Amazing.
Yeah, great. Thank you.
Well, I think that's revolutionary.
Yeah, our artigraphy group is really excited about this.
Other questions?
There's a comment in
Is
the
that chat?
How much does a BioBox cost, not including shipping?
Well, I should know that, but you mean to put together? Not much. As these things go, I'm just off the top of my head, $10.
Wow.
That's just for the box, right?
No, 20. No, that's for that and the supplies. The most expensive thing is the bio booklet. A lot of effort went into that, and we have it printed on really good paper, cardstock, by a very talented printer with a great graphic designer who has helped us. And I think that's key, just having the kind of instruction sheets that we're used to following just do not fly with people who don't have a high school education. And again, we're trying to get everybody in our study, no matter what.
Any other?
I'll send you the cost. That may well be.
Seems low.
I was able to buy all this. I mean, and Hannah and Jocelyn, research assistants, we bought the stuff off Amazon, the veterinary tape. You know, I got a deal for $17.99 for more tape than we'd ever need because it's used for the animals. You know, so.
Any other questions?
Oh, the watches are expensive.
Yeah.
The gene active are, but they can be recycled.
Yeah, about
So
200.
they return those to you then in the box?
Back in again, and there's a cost to doing that. So
Yeah.
that's not cheap. And of course the assays are not included in that cost. But the goal was, originally we thought, you know, plastic molded, you know, all this kind of stuff. And this was a good enough first pass. Now that we go into the main, I don't know if we have time for that. Personally, I think it's more appealing if it's sort of loving hands at home, you know, in Ziploc bags with stickers. It's like more personal. They're helping us, the investigators, and contributing to science. But that's my science for the people background.
Martha, we have one other question, whether you'll be publishing a methods paper on this?
People say I should.
I think that I would agree. I think if people would be, there would be a lot of interest.
Yeah, well, we absolutely are planning on doing that, and especially will as we consider increasing the proportion of the sample in our main wave 4 sample. But my goal here was to get you compliance and scoreability data on everything. And that's what we focused on doing. Now we're going to do the hard work of actually scoring absolutely everything. And then doing the validation and standardization studies, some of which we've done, and they're good, or we wouldn't be doing this. But there's still some that need to be done. I have a question for everybody if we have time.
Yes, we do.
Notice there's no blood. There's no blood, and therefore we're missing our key measures for the most common mechanisms in morbidity and mortality, namely diabetes, chronic inflammation. We want to get COVID antibodies. We have HPV antibodies. And so if anybody has any experience with these new micro samplers, which have just blossomed in the past year, and then multiplex assay companies that we can work with to get all of, 6 or 7 analytes. The ones I've looked at right now is that you can get 67 analytes out of, 2 ml of blood, but you need to marry the collection device and the assay. And dry blood spots, we tried and self-administered. It's just, I'm preaching to the choir here. I would say something's better than nothing. and Tom McDade says he can answer that question. So maybe
good for Tom.
Yeah, well, I can provide some input. I don't know if it's going to be an answer, but with respect to coronavirus antibodies, we have a blood spot method that works really well, and we're embarking on this experiment of at-home collection like everyone else, just out of necessity. And I've never been a fan, as you know, of the at-home collection just because the quantity and quality of sample isn't as reliable. But it's a question of getting something or nothing. And so we just launched the study in Chicagoland area. We're aiming to recruit 3,000 people. We have a web interface. So they enroll through the internet, they do a survey, they consent, they give us their address, and we send out a collection kit. So we'll know in a couple of months the quantity and quality of sample that we get. For the antibodies, we don't need much.
Doing the dry blood spot on
the dry blood spots, yep.
Oh, great.
And how many spots are you asking people to do, Tom?
Well, we're asking them to give us 5 and then fill out the card. We'll see. We only need 1 small disc to do the antibodies. For the multiplex cytokine approach that Martha was referencing, we have a good MSD protocol that does IL-6, IL-10, and TNF-alpha, but it requires 1 full punch of blood. In reality, it tends to require 2 drops of blood. I'm sorry, it requires 1 full drop of blood. So it really requires 2 because most of the drops that come back aren't full. I've looked into the Mitra devices, the Neoterix. You know, they look like a Q-tip with a little
matchsticks, I call them.
Yeah, matchsticks, and they're pretty clever. It's and when I first saw it, I thought, oh, why didn't I think of this? But and one of the main advantages is that it collects a fixed volume of blood onto the tip. So then the processing in the lab there's some steps that are easier because you can just drop them into a plate to elute the sample. The problems with them, and the reason why I haven't decided to turn to them, is that they're expensive. You know, a filter paper costs $1. These devices cost anywhere from $10 to $30, depending on volume and the device. The sample volume is also quite limited, so each tip will collect either 5 or 10 or 20 microliters of sample. You can get them in different sizes, but when you get the device, you only get like 2 tips, or maybe you have to get another device to get 4 tips. So the volume of sample that comes back to you in the lab is greatly reduced. They also cost a lot more to ship because they're more than 1/4 inch thick, whereas the filter papers are flat. So we looked into them for our coronavirus antibody thing in Chicago, and it cost us $3.80 to send out a collection kit, but it only cost us 55 cents to return a sample because the filter paper goes in a flat business envelope with a little desiccant. Whereas the Neoterix clamshell device would cost about $3.80 to return. So for those reasons, we haven't pursued it. But lots of people are using it. It's being used in a big NIAID coronavirus antibody screening project. So it's out there, and I think for some applications it's worth considering. But for my purposes, it hasn't worked out yet.
Right. So, John King, do you want to ask your question yourself?
Sure, it's more of a request than a question. It was great to hear from Tom what experiences that they've been having with coronavirus testing, but needless to say, we're getting asked by everybody, how best they should do this. And we don't really have an awful lot to tell them. We can show them the page of standardized tests and things that seem to be in approval mode. But in terms of reports from the trenches and what has worked, or the kind of thing like, for example, if you're doing it by mail, it's going to cost you 8 times as much to use this one than that one. It's not something we would have picked up. It's not any of the, in other words, basically we're looking for the lore because everyone wants to do this and makes a lot of sense for a lot of the studies. But at the moment it's early days and we really don't know what to tell them. So any advice you could, any tips you could hand back to us would be really great because there's literally hundreds of researchers who want to do this.
So perhaps we should get a webinar on doing just this. Going
next
month or so. It might be helpful because everybody's in process.
I would say, if I could just add 2 comments. One is, like I said, like probably in a month, we'll have good data on the feasibility of self-collection and how, the quality of sample that we get that's usable in the lab. The second point I would make, if you're thinking about going this way or in this direction or funding in this direction, is that, there are a lot of people who are assuming that if you test for an antibody, that confers immunity. But all of the tests right now are for binding antibodies that are, strong signals of prior exposure. Well, we know nothing about what that means for subsequent immunity. So it's more of a retrospective tool in terms of where the virus has been and who has engaged with it and developed a response to it, not a forward-looking tool in terms of immunity. There are some methods that we can develop to look at that question, but that's not where antibody testing is right now.
No,
Yeah,
that's
I totally agree. And I have been struck, and I think in reporting it, because people want to get reporting, that's what you have to be really clear about. But there are assays now for neutralizing antibodies,
Right.
and it's IgG3 that matters. That's the best, indicator. And there's one group that does 2 spike antibodies, and therefore it has 100% sensitivity. But this whole field is just moving, brand new. I mean, science at rocket speed. So,
yeah.
Well, there does seem to be a lot of interest in a possible webinar. So Eileen, you and I can connect up and see if we can get something organized.
Yeah, and it could be mainly chatting with people who.
Yep.
That would be wonderful. We, if we could find a couple dollars to throw into that, we would. I don't know if we can, because we're throwing them all at people actually collecting. No, really, every
we can do webinars without a few dollars. We don't have to put it on charge.
We can get the word out and ask people to come, or be prepared to say a little bit about anything they're working on, or bring their questions.
We'll come back to you and ask for suggestions of people that are testing. Because I mean, we know some individuals who are testing various things and pros and cons and approaches. So, and we'll include Tom and Martha.
Yeah, no, the biggest effort that's going to be ongoing, much sooner than I can even imagine, is going to be all the radix up testing efforts that we just put the stuff out in the street for. I mean, that's going to be a very, very, very big high profile project that's going to need to figure out a lot of things really fast. And this is one of the simpler things actually, but but if there is an answer, at least we'll know it, and that would be really great. So thanks very much. And I'll mute myself again because I know you've got many other fun things to go here.
And let me just say, my email, I should have it here, is a Yahoo account. Try to reach me at UChicago for a variety of reasons. So it seems weird, but that's what I use. So I guess there's a list, isn't there?
We can circulate.
That would be great, because I'd love to email with a bunch of you, or Zoom or whatever.
Any other questions?
I don't see anything.
I don't see anything in the chat for the participants right now.
No, I think we're good.
Okay.
So I think we're coming to the end for today. Just wanted to remind people there is our 3rd and final session on Monday, and that will be focusing much more on methodology issues, and particular some updates on a project that's been ongoing around methodology related to telomere assessment, collection and assessment of samples. So hopefully you can join us. This session and the next one will also be recorded and posted. So if you or your colleagues are unable to join, you'll have an opportunity to take a look then if you want to. And the one from last Monday is posted, I believe.
It's on the homepage of our website for the Center of Biodemography and Population Health right now.
And thank you all. This was another wonderful session. Thank you to all the presenters. These were just terrific. Thank you so much.
Thank you so much, everyone. And this is a great group.
Have a good weekend.
Nice to see you all.
Hopefully we'll see you on Monday.
Thank you all.
Bye.
Bye-bye.